Why is the study of entomology important?
So, entomology and the study of insects is good because through work with Drosophila, mosquitoes, other insects, we see a lot of similarities between the invertebrate world and the vertebrate world. And so a lot of people study insects, and that information can parlay into human studies. You don't have the restrictions that you do with vertebrates, especially mammals and humans, in terms of animal use protocols. Insects are a much simpler model as well. Their nervous system is very similar to humans in terms of how the neurons work, in terms of the neural physiology, the neurotransmitters present, but you can dissect an insect. You can raise hundreds or thousands of insects at a time and have a large sample size as well. A lot of people like to study mice. If they don't study humans, they study mice. The generation time for a mouse is up to 6 months or so. You can get new insects, new adults, in about 10 days. And so there's also just an ease of doing the research and growing the organisms. And besides the fact that due to the size of insects, you can also house hundreds or thousands of them at a time and not take up that much space.
What are eusocial insects?
Eusocial insects are insects that live in colonies and that they work toward the betterment of the colony, not just the individual. And so examples are ants, bees, termites. Most people are aware of bees and ants being eusocial, but termites are also eusocial. They're typically in castes where there's a queen at the top of the hierarchy, and she's the only one that is reproductively fit in terms of females. And then there are workers and soldiers who are sterile. They can become fertile when the queen needs a mate to repopulate the colony. Otherwise, they are in charge of foraging for food or defending the colony.
What are the origins of eusociality?
Eusociality is probably derived from one of two theories. It's either that the relatedness of the individuals. So typically a colony shares a lot of genetic traits because you have one queen that is reproducing for the colony. And so one benefit is that even though the workers are sterile, we would say that typically they would be unfit because they can't pass on to their offspring. They can't pass on their genes. However, because they're all related to each other, a benefit of eusociality is that you are still working toward the betterment of the colony and the genes in that colony. And so workers are more related to their sisters and the queen than they would be to any of their offspring. And so one benefit is just that you continue to propagate your set of genes, but you've now divided the labor of the colony, and survival is not dependent on one mate alone. It is a group effort. And so there's a lot of protection and foraging that can be done in the absence of needing to actually find a mate and reproduce.
Well, there have been a lot of theories put forth about the evolution of eusociality. Part of it has to do with the ease of development of eusociality and why it might appear in several independent lineages of Hymenoptera is that Hymenoptera are haplodiploid. So the males are derived from unfertilized eggs, and so they're haploid, and females are derived from fertilized eggs, and they're diploid. And there are a number of theoretical issues that suggest that haplodiploidy is facilitates the evolution of sociality. And of course, that's not true for termites, which are not haplodiploidy, but in termites, the evolution of eusociality is thought to be a single evolutionary event, whereas in Hymenoptera, the evolution of eusociality, as I mentioned previously, has occurred independently in several groups.
Pheromones used by eusocial insects.
So in the ants, and ants have a variety of different pheromones that are used for foraging, and so similar to bees, they might have recruitment type pheromones, but they also have foraging pheromones, so that they lay down, as they're foraging, many ants will lay down trails that allow them not only to return back to their nest, but also for other foragers to follow up the same trails, particularly if they're successful. So they can lay down, if they find a food source, they can lay down pheromones that suggest successful foraging. That allows recruitment of other individuals, but also allows increased numbers of individuals to find the food source, and they can do that very rapidly.
In order to study eusocial insect foraging, a mathematical model using differential equations was developed to model the communication mechanisms that produce complex and functional group-level foraging patterns for the two species. Based on a consolidation of studies mentioned for honeybees, Apis mellifera, and termites, Reticulitermes flavipes. Honeybees can be characterized by many different aging phenotypes. Based on these phenotypes, a framework of behavioral states where different workers follow different roles outlines the foraging in Apis mellifera. Followers are made up of honeybees attempting to follow their recruiters to the food source, or those conducting independent search for food after being given a signal. Searchers are honeybees that initiate foraging activities, get lost after following or exploiting, and continue to search for food. Exploiter honeybees collect the food and do not directly recruit, but do leave a pheromone trail for others to follow later. Recruiter bees have the task of attempting to recruit nestmates and actively leading workers to the food source. This observed behavior simply defines the behavioral states and equations that will illustrate the allocation of populations according to the number of honeybees at the given task. Exploiting honeybees exhibit an exponential growth as honeybees from other behavioral states join this state. Ideally, in nature, the main point in foraging communication and mechanism is to ultimately find and obtain food. Here we have the following honeybee graph, which shows a growth of workers as they stop waiting and are providing instruction by the recruiters to commence their journey to the food source. The slope follows the ideal pattern in nature, as follower populations should increase with increased interaction from recruiters. The number of recruiting nestmates decreased as time went by. Honeybee recruiters only have to recruit initially. Thereafter, the new bees begin following and foraging. The assumption follows that the effect of the initial recruiting bees gradually decreases as a pool of following and exploiting bees increases in long-term steady-state behavior. The searching population is dependent on the rate of recruitment from the waiting population. The graph illustrates a linear result for the searching population. When searching begins, time spent returning to the nest decreases if food is found and exploited, so the overall rate is increasing as the recruiting threshold is met and departure from the nest continues over time. This figure shows the waiting honeybee population at the nest as it decreases over time. The greater the waiting population, the more rapidly it decreases in numbers. Colony-level patterns should exhibit similar behavior because an increased number of available foraging population increases the number of individuals performing in behavioral states, progressively increasing the rate at which foraging signals communicate information to other foraging bees. This pattern is similar to the exploiting and recruiting patterns that depend on the activation or deactivation of other behavioral states to emerge with a growing number of individuals to perform their state function. Trail pheromones are the primary axis of communication for laboring ants and termites. The mechanism of chemical communication is similar in both of these species, and experimental testing using ants suggests a connection in recruitment behavior. Based on those studies, a model was designed to apply the different pheromone concentrations and see how the pheromone affects the foraging rate over time at an optimal population of termites. The termite population activated by the pheromone trail eventually reaches a plateau as the continuous and repetitive passage of termites accumulates the optimal pheromone concentration for efficient communication, this being 10 femtograms per centimeter. The graph begins with the pheromone threshold of 0.1 femtograms per centimeter and increases until it reaches the plateau or gradient of activation. The results obtained require experimental verification with population data from both honeybees and termites to verify the patterns being observed in these theoretical applications. Comparing honeybee and termite foraging is a bit problematic due to the different influences that produce foraging. Curiosity in the collective foraging that emerges from an individual in eusocial insect societies has always posed question about the dynamics of the organizational pattern that brings forth great success in their populations. To ponder about the interactions hundreds of thousands of members contribute to an overall framework in a highly proficient society is beyond grasp. Mathematical models offer a hold on perceptions of the simulation of various parts of this framework to better comprehend the atomistic nature of eusocial insect societies. What better way of studying insect ethology while incorporating equations that described insect behavior to help us appreciate their spectacular nature, evoking nothing less than admiration and enrichment from their majestic contributions to this earth.